Hello friends, welcome to BMH Learning. In this video, we are going to discuss about another very important signaling pathway which is known as IP3 DAG pathway. From our previous knowledge, we know following the ligand binding activation of this GPCR, the G alpha subunit detaches itself from the receptor as well as the beta gamma subunit of the trimeric G protein, and then it goes and binds to the effector protein here, as we saw in case of the CAMP pathway where the effector protein was adenylyl cyclase. In this case, the effector protein is PLC, that is phospholipase C enzyme. If you do not know how it happens, you can watch the previous parts of this lecture series. Now this PLC enzyme or the phospholipase C is not solely activated by only GPCRs, but there is another pathway which involves tyrosine kinases. These receptor tyrosine kinases, they also activate this PLC enzyme, but we will not that discuss now. We will talk about them in the future videos. So now what is the function of this PLC enzyme? It stands for lipase. So if it's lipase, it means it would act on lipids. More importantly, this is phospholipase. So that means it acts on phospholipids, and when it does so, it converts those phospholipids into some other compounds by breaking down the phosphodiester bond which are present in those phospholipids. Here this enzyme is acting on IP2. This is phosphatidyl inositol diphosphate, bisphosphate, which is embedded into the cell membrane. This is a derivative of a very important molecule which is known as PI, phosphatidyl inositol, and this molecule is always present in the cytosolic leaflet of the plasma membrane. So we get this PIP2 from PI. Now the action of this PLC on this PIP2 produces two another second messengers which are known as IP3 and DAG. Okay. Action of PLC on PIP2 is producing two different compounds which are known as secondary messengers. They are here IP3 and DAG. Talking about DAG, this is diacyl glycerol. So this diacyl glycerol is a lipophilic molecule. It says, I love lipids, so I am not going to go anywhere. I will stay only here in the cell membrane where I can find lipids. And this IP3, phosphatidyl inositol triple phosphate, this is a polar molecule or hydrophilic molecule, so this is not going to stay here. It says, I will go somewhere else where somebody is waiting for me, so it diffuses from here. Now we shift our focus from here to something else. This is the figure of endoplasmic reticulum. Okay, on this figure, this is the membrane of ER, and on this membrane we have green structure here. What is this? This is a channel. Channel means a protein which allows the transport of some minerals or ions through it. Specifically, this is a calcium channel, calcium ions channel. So it allows the entry or the exit of calcium ions through this channel. Then we have gated channel. So it's a gated channel. That means sometimes this channel is closed and sometimes this is opened. Now for the entry or the exit of these calcium ions, it needs to be opened. How it would open? This is a question. For this channel to be opened, there is a condition, and that is the binding of those molecules for which this channel has been designed. It would open only in that case when those molecules would bind to this channel for which this structure is designed, and that is the IP3 which we just synthesized. So this is IP3 gated calcium channel. So we are calling now this IP3 molecule from here to here. So we have here IP3 molecule. Before the binding of the IP3 to this channel, this is closed. Now they will bind to this channel protein here, and the channel is opened. After its opening, the calcium ions would go from here to the cytoplasm of the cell. Right? Now the calcium ions are present in the cytoplasm, and in the cytoplasm we have an enzyme which is known as PKC, protein kinase C. This is in inactive form. Okay, so the calcium ions, they bind to the PKC, but the binding of the calcium ions to the PKC is not making the enzyme active. The binding of the calcium ions is not resulting in the activation of this PKC enzyme. Instead, the function of these calcium ions is that they bind to the PKC and they take this enzyme to the cell membrane. Or in other words, the calcium ions recruit the PKC to the cell membrane. Okay. And after the PKC is present in the cell membrane, what next? Remember this DAG? Now this would come into play. Diacylglycerol is going to do its action on PKC and makes the enzyme from inactive to active. So if you notice clearly, the activation of the PKC is the joint action of both these IP3 plus DAG. How? Initially the IP3 goes to the closed, this structure, the channel, and helps in the exit of these calcium channels, these calcium ions. These calcium ions, they bind to the PKC which recruit the enzyme to the cell membrane, and after that the DAG, because it's a lipophilic molecule, it cannot come here into the cytoplasm, it has to be here, so it acts on this enzyme and make it active. After the activation of the PKC can go on its substrates and phosphorylate them, and then we see the results.